To me, the question quickly became one of, are we going to try to conserve institutions, or are we going to try to conserve diversity? Because the two are not the same. I realized that we had 1,700 institutions out there conserving genetic diversity, and if we set up an entitlement program, I would get 1,700 pieces of mail saying, we've got seeds, you send money. And even more alarmingly, when the roof started to leak on these 1,700 facilities, we would get calls in the middle of the night saying, the roof is leaking, please send money. I realized we couldn't do that because if you're the executive director, as I was, of an organization that's structured as an endowment fund, you quickly begin to do the math. And if your endowment fund is set up as ours was to yield 4% a year, let's hope it's 4, then if somebody hits you with a $40,000 bill, which a lot of my staff at the time thought was something trivial, you actually have to raise $1 million in order to meet that $40,000 bill every year. So you become very focused on what you're going to do, and more importantly, what you're not going to do. So our strategy was essentially derived by arithmetic, not brilliance. We wanted to create a global system, and we wanted it to have these characteristics, which sounds a lot like mom and apple pie, rational, sustainable, cost-efficient, and everything. But each of these words actually does mean something. We decided we would focus on unique germplasm, and we would try to facilitate it being conserved where it was best and cheapest to conserve it, consistent with maintaining the quality of conservation and maintaining availability. But first, before we could endow that global system, we had to create a global system because it really didn't exist. And when we looked around at the unique germplasm in these small collections that were being run in poorly funded and poorly staffed gene banks, we found a real disaster. We found a huge amount of diversity that was in danger of being lost quickly. So we engaged, we started working with over 70 countries to identify and essentially to rescue the threatened diversity within their collections. And over a period of 5 years, we literally rescued from extinction about 75,000 different unique crop varieties. It's what Peter Raven called one of the largest biological rescue programs in human history. Having rescued it, we of course didn't know much about it, so we also engaged in a number of evaluation projects where we characterized and evaluated the material so we could at least know the basics about what it was we had. Then we began to try to articulate what it meant to have a global gene bank system for agricultural crops. And quite simply, we thought it was a three-legged stool. It was each individual unique accession being stored in 2 collections, 2 normal working gene banks, plus backed up in a fail-safe facility, which was the Svalbard Global Seed Vault. Now, why should it be in 2 working gene banks? Well, because bad things happen to gene banks. And second, because of the politics in this field, one can't simply, I'm sorry to say, count on every single gene bank actually offering access to its material. So we hoped that having 2 would solve that problem, backed up in Svalbard. This system, of course, is based on the assumption that there's some cooperation in the world, and that may be an assumption that that does us in someday, but at the moment, that's what we're counting on. Now, I want to take a little bit of time in introducing you to Svalbard, partially because it's maybe the least known of what I'm talking about, but also because I've been very involved in it and I happen to love it. So I want to give you a very, very quick tour of the seed vault, and then I want to spend the last few minutes of my time with you talking about the future of gene banks. This is where we're talking about. This is the farthest north that you can fly in this world on a regularly scheduled airplane, group of islands called Svalbard. That's mainland Norway below. So if you're in northern Africa and you fly to Oslo, you're about halfway there. Takes you quite a long time to get up to Svalbard. It's governed under an international treaty to which Norway has sovereignty. It's a remarkably beautiful place. I just spent about 10 days up there a couple of weeks ago on a boat going around the group of islands. Yes, polar bears, walrus, seal. It's just it's an amazingly beautiful place. This is a glacier. If we were standing at the foot of this glacier and looked up, you'd be looking up about 300 yards. It's a huge, huge glacier. Breathtakingly beautiful. This is the facility we designed working with engineers and architects. The idea was to place it, first, the idea was to place it in Svalbard. Why? Because it's a stable place. It's overseen by Norway, which many countries trust. It's a demilitarized zone under international law, and it's very cold. And so if you want to save seeds for a long time, you freeze them. And since no mechanical system is perfectly reliable, we wanted to go to a place where we got free freezing, at least down to the point of about -5, which is the cold spot inside the mountain, inside the permafrost, -5 Celsius. So this was the design that we worked on, of a tunnel leading back to rooms inside the mountain, to the coldest place in the mountain, which would offer us the free permafrost and the free freezing for our seeds that would be safety duplications of big gene bank collections. To give you a sense of scale, this is the seed vault under construction. And this is the seed vault today in situ. Now, when you open the front door, you're going down a tunnel. A lot of people think they're going to see some pristine laboratory conditions, but no, this is very practical. It was financed completely, 100% by the Norwegian government for the cost of $9 million. I like to say it's the cheapest insurance policy in the world. So you enter, you go down this long tunnel, about 130 meters. You go through a number of doors that increasingly look like this because it's very cold. And then you open that door and you get into the first of 3 rooms. This room today holds seed samples of 783,000 different crop varieties. This is the world's largest collection of agricultural biodiversity, maybe collection of any kind of biodiversity in a single room. There are over 100,000 varieties of wheat and 100,000 varieties or populations of rice in this particular room. The way it functions is that depositors, their institutions, will send us a duplicate copy of their seeds under a standard deposit agreement. They retain ownership of the seeds. We simply will store them for free, essentially forever, to the best of our ability. The forever part is where the endowment comes in, because we generate funding on a permanent basis, and we fund the operations of the seed vault, which, by the way, are rather inexpensive. So we're talking about annual operating costs of less than $200,000 a year for the largest gene bank in the world. But so it's a fairly simple procedure. It provides the best conservation facilities in the world. None are better. So in other words, the seed inside the seed vault in Svalbard don't deteriorate any more quickly than the seed in the best gene banks. And what that means is that we don't have to do testing and regeneration and such in Svalbard. We simply allow the owners of the seed, the Fort Collins and the different gene banks of the world, to do that. And when they realize that some of their samples or accessions are in need of regeneration, multiplication, they perform that. It's their duty as a gene bank to perform that, and they send us another copy up in Svalbard. We have 3 rooms this size. Like I said, we have 783,000 samples in this room. We have a capacity of 4.5 million samples, which I think is about 3 times as much as exists in the world today. But we planned everything about the seed vault. It was planned with some redundancy. And since I was the one that made the estimate of how many unique samples there were in the world, I built in a bit of margin of error, knowing myself. I guess one of the things I would like to say about this seed vault is that it is a very special kind of experience to walk in it. And even though I spent a lot of time in this seed vault, I never fail to be touched emotionally when I walk through this room, realizing what it means, what it represents in terms of the history of agriculture, what it represents in terms of the future of agriculture, and my sense that finally, knowing what has happened to so many gene banks, even in recent years, Iraq, Afghanistan, Solomon Islands, lost. Finally, when you go into this room, you realize, well, we've got at least this portion of agricultural biodiversity that is, as far as we can tell, will be safe forever. Quite an amazing thing, and my hat's off to Norway for allowing us to do it there. Now, I want to turn to the future for a second. And as someone who is at the much closer to the end of their professional life than the beginning, I suppose I'm allowed to throw out some wild ideas, and I want to throw out 1 or 2 for you to domesticate today. We know that the future is going to be a future in which agriculture experiences severe water constraints. Agriculture already takes 70% of fresh water supplies. We're competing with industry and urbanization. We're probably not going to get more than 70%. There are nutrient constraints. Phosphorus production is slated to peak in this century. I've already spoken about land constraints. And then, of course, there's climate change. Here is a chart showing what percentage of the overlap of future temperatures with present temperatures in Africa. And you can see that later in this century, there'll be virtually no overlap. So if you're an agriculturalist, you want to ask yourself the question about the crops in the field today. The crops in the field today that are adapted, so to speak, to today's climate, are they going to be equally well adapted to that climate on the far right of this chart when the temperatures don't overlap at all? So what about those hundreds of, I would say, dysfunctional gene banks in the world? The gene banks that have poor funding, that aren't distributing any material, that have poor conservation standards. What are they going to do? And are they going to get funding? Who's going to want to fund those kinds of institutions in the future, of which there are many? And we're talking about many, many national programs, particularly in developing countries. And what about the farmers? The farmers that are going in Africa who are going to experience what you see on the chart here. There are about 1 billion farmers that live, about 1 billion people that live in the world today who live in farm families that are largely self-provisioning in terms of their seeds. In other words, they're outside of the orbit of both the private and the public sector. So adaptation, crop adaptation for the poorest of the poor, for those 1 billion people, boils down to the genetic resources they have on their farm. Now, you want to ask yourself, are those genetic resources on the farm, at the community level in Africa, sufficient in terms of containing the traits that are going to be needed for adaptation for a completely different climate? I don't know the answer to that, but I would argue that it's unlikely. So here's crazy idea number 1. What if we were to look at our gene bank holdings and work with climate scientists, begin to model the types of climates that are coming? What if we were to look at our gene bank holdings and begin to assemble the traits that might be interesting or necessary for the future? Our best bet, no crystal ball here, but our best bet. What if we were to try to begin to put together the land races, the accessions, the populations of these types of materials? Stuff like, I think I have a slide or two. Here's one. This might look like 7 different varieties of wheat. It's actually a population collected in a single farmer's field. So this is 1 sample in a gene bank, not 7. What if we were to put together material like this, combined with some maybe seeds of modern varieties with interesting modern yields associated traits? And what if we were to begin to use those small gene banks to do the testing and to oversee some distribution and to distribute small packages of seeds to farmers around the world, people not touched by commercial or private sector or the public sector. Small packages of seeds, not enough to plant their whole field, but enough for them to test out and experiment and integrate into their farming systems if appropriate. So that we would turn millions of farmers into plant breeders and experimenters, people who otherwise are not going to be touched and are not going to be armed to adapt to climate change. What if we were to do something like that using the institutions which today are neither conserving nor providing genetic resources, and there are hundreds of them? Well, this is a crazy idea, but it's actually not my idea. This is an idea that we all have experience with in this country because this is how we got American agriculture. Back in the 1800s, 17 and 1800s, as I mentioned, the precursor to the U.S. Department of Agriculture, which was the U.S. Patent Office, in recognition of the fact that agriculture was an art and a science, and an innovative one, began to assemble seeds and planting materials, and it began to distribute them all over the world, all over the United States, in small packages. They had learned how to put seeds in packages because they had looked at the Shakers, who were the first to start seed businesses and put seeds in packages and sell them. And they began to distribute these packages of seeds all over the country in experimental quantities. This is how you have wheat growing in North Dakota, which is a crop native to the Near East, because you turned farmers in the United States into experimenters and plant breeders all over the country for different types of crops. And this is the only way you can explain how all of our different crops came to be grown all over these United States.